Hello, chapter 13, part 3. Here we go. Back to blood groups. With blood groups, you have something called an antigen, which is the marker. And the marker is found on the blood cells. Type A has an A marker. Type B has a special type of marker called B. Type AB has both the A antigen and the B antigen, and type O doesn't have any sort of antigens on it at all. Now, in the plasma is the antibodies, or the anti-whatever bodies. So the type A, as you see here, has your type B, anti-B antibodies. Type B has anti-A antibodies. Type AB has no antibodies, and type O has both antibodies. So, in order to have agglutination, which is a fancy word for clotting, you must have an antigen on a cell reacting with the same type of antibody. When it comes to donating blood, you can always donate blood to the same blood type because it's the same. But if you can't, you have type O because there are no antigens. So type O blood is seen as the universal donor because there's no antigens to cause any problems with the antibodies. Now, when it comes to being the receiver, type AB is the universal receiver because there's no antibodies to cause the reaction. Again, in this situation, instead of memorizing this whole chart, think of it logically. If you have an antibody on your cell that interacts with an antigen in your receiver's or in your donor's body, you will have agglutination. O is the universal donor because there's no antigens, and AB is the universal receiver because no antibodies. Kind of confusing, but hopefully you can sort it out. Good. Okay, so there's also another marker called the RH factor, and it's called RH because it initially was found in rhesus monkeys. If you have the RH antigen, you are said to have positive. If not, you are negative, and this builds on top of the AB, and O. Now, this can be a problem only if there is, I don't have a diagram for that one, sorry. If there is a father that is positive and the mother is negative. And because it is, the RH positive is dominant, the baby will inherit the positive factor from the dad. So the mother has no contact with the positive, but as soon as the blood interacts for any reason, it could be bleeding at the placenta or it could also be during birth, the mother will develop antibodies which will destroy the RH positive fetus, usually in the second situation, not usually in the first. So you must have certain conditions. You must have the father, which is positive, the mother, which is negative, and blood has to mix for the mother to develop antibodies against the positive. Now, there is also a shot to destroy this. So this is why if the situation arises, you can have a shot to destroy the antibodies. Okay, so it's very specific, but it is quite deadly if that right combination arrives. And it's called erythroblastis fetalis, just because I want to throw out that big word for when you see this condition again. Blood clotting, last thing I hope. No, it's not the last thing, it's second last thing. Blood clotting, it's very important to prevent loss of blood and loss of blood volume that we have a process that clots. But we only want clotting when we want the process to stop. You don't want extra clotting, or then you have a condition where you have clots floating in your blood, and you don't want clots floating in your blood. Excuse me. You don't want clots because they'll clog arteries. Now, This is a very complicated process with over 12 clotting factors, but we simplify it, and there's also a video to happen. So there's an intrinsic and an extrinsic clotting factor. So this happens when the platelets are activated. Now, notice these platelets are drawn a lot larger than they are. When there's an injury and damage to the clotting factor, damage to the vessel, the clotting factor will cause the platelets to be activated. Once the platelets are activated, they will stick together to form what's called a platelet plug, which you'll see in the video. And then the platelets will release something called, well, they release 12 different enzymes, which eventually gets to something called prothrombin. Prothrombin will activate thrombin, and thrombin is an enzyme acting on a fiber that is, or a protein that's in the blood called fibrinogen, and that will cause fibrinogen to be turned to fibrin, and fibrin will then form these stringy mesh inside, which the blood cells get caught in, and you have a blood clot. So it's actually quite a very complicated process, but we simplified it in the fact that platelets get activated. Platelets result in forming a platelet plug to temporarily block things. Platelets will also release other chemicals, one being serotonin, which causes the vessels to constrict. Another is to activate all the clotting factors, the one being prothrombin. Prothrombin, in the presence of calcium and other clotting factors, will turn it into the enzyme thrombin, and thrombin changes fibrinogen to fibrin. Fibrin forms a mesh, blood cells get caught in it, and you have a scab. Very complicated, but try and get the general gist of it. And this is very important because it's very important to stop the bleeding to keep our blood volume up. There's all sorts of situations where clotting goes bad. I want you to realize that you can have a thrombus. A thrombus is a clot, and an embolus is a moving clot. And both of them are bad because you don't want a clot or a moving clot to clog an artery that you don't want clogged. Okay? There's all sorts of information here. Now, when we look at blood vessels, we've mentioned this before in a bit. Let's look at it here in terms of, there's a whole bunch of names of blood vessels. You don't need to know the names of these blood vessels. That's so next course. I want you to be aware that there are three types of blood vessels: arteries, capillaries, and blood veins. Arteries, capillaries, and veins. Now, an artery will start large and move to a small artery called an arteriole. Arteries are under high pressure, so they're very muscular. Both arteries and veins have three layers: an outer protective layer, an inner smooth layer, and then a middle muscle layer. You don't need to know the details on the layers and all the names. Just be aware that there are three layers. You also want the inside to be very smooth, so that way it doesn't activate any clotting. So the arteries are very large and very muscular. The veins, on the other hand, do not have such a muscular layer, and they also have valves to prevent blood from going backwards. The capillaries are the vessels where the transport into the fluids occurs. Into the tissues occurs. Capillaries are only one cell thick, and because they are so thin, this is where fluid exchange happens and gas exchange happens. All the other tubes are just for transporting. It's in the capillary network or the capillary bed where the exchange occurs. Again, the textbook goes into a little more detail, so let's briefly look at these. Blood leaves the heart through the aorta, and which is the main transporter. You'll see that if you saw one of the videos. Arteries then divide smaller and smaller, and they become to arterioles. The arterioles will fill the capillaries, and it's in the capillaries where they're single cell and things can diffuse or be pumped across. Then the blood is collected in the capillary and then will go into the venule, and the venules will eventually get larger to veins and go back to the heart. There are 3 layers. You don't need to know all the detail other than there's an inner layer, a middle layer which is muscular, and then the outer layer which is usually used for protection. Vessels change depending on their job, as we talked about it. Arteries are thick-walled, veins aren't. Capillaries are very thin. Nutrients move back and forth in the capillaries, and you have millions and millions of capillaries. We talk a little bit about something called capillary beds and precapillary sphincters. When we talk about the sympathetic nervous system shutting it down, it shuts it down, shutting down a substance such as in the digestive system. There's these precapillary sphincters which will close and shunt the blood somewhere else. So if they're open, blood travels. If they're closed, blood is shunted somewhere else. And that just is a way to put a lot of blood into the area. So if your muscles are contracting, you want a lot of blood to go to your muscles. If your sympathetic nervous system is taking the blood away from your digestive system, you're closing the sphincters. Blood pressure. Blood pressure is basically measures systolic pressure over diastolic pressure, and goes into a lot of detail, but we're already getting far too much detail here. So just realize pressure is a measurement of the blood pressure is a measure of the pressure in the blood. Good. Now, controlling blood pressure, the blood pressure is controlled by sympathetic nervous system. Sympathetic nervous system will cause the blood pressure to speed up, usually by vasodilation, and vasodilation makes the hoses smaller. You also have hormonal control, and we'll talk more about the hormonal control when we talk about volume and urination. Now, the last bit is the lymphatic system. When we look at the lymphatic system are the green tubes, and they're basically a system that runs parallel to the circulatory system and collects extra fluid. We'll talk about that in the next, not the next unit, the following unit on the lymphatic system. There's a lot of stuff on disease. Have a look over it so that you're aware of it, but just generally, usually you're having a problem pumping or not pumping, pumping too much or too fast. Then you can also have a valve problem, which clogs the valves or clogs the veins. And then you have a blockage. So there's different categories there, but far too much to cover in this class, so we won't be I won't be quizzing you on the problems, but I would like you to read them over just to be aware of them. And that is part 3 of a very big, very complicated chapter. So don't rush it. Take your time. Please make sure you watch all the videos to sink in all the information. Thank you and happy learning.